Thanks, Christian, and thank you all. And truly is a great pleasure to be here. And thanks for scheduling my talk to be the last day of the talk because I'm still on U.S. time. So yeah, okay. So by now I hope I'm up. In any event, yeah, what I'd like to tell you today about is group dynamics and phototaxis and modeling. I try to be light. I'll try to give you some ideas about what we are doing. But before that, yeah, just I noticed that, United States is a big country. So when we say University of Maryland, not everyone knows where Maryland is. So why not start with some, that was not supposed to show there, but okay, with something light. So let's see. This is the ocean, the Atlantic Ocean. Then that's the east coast of the United States. Yeah. Here's the Chesapeake Bay. And that's the Potomac River. And that side of the Potomac River is Maryland. The other side is Virginia. And if you zoom in, let me do that. You zoom in, you see the city of Washington. Washington initially was a diamond, but Virginia took half of it. So it's actually like half a diamond starting from the river. And the rest is Maryland. That's Maryland, and here it's Virginia. So if I zoom even more, it's more interesting than the talk, right? Okay. So you have here the main area with the museums and the Capitol and the White House. And if you're looking for the University of Maryland, basically what you have to do is you look a little bit here. That's College Park, Maryland. So this is 12 kilometers from this, so just give you a scale. This is the beltway that is around the city. And there's very interesting trivia fact about this beltway. And that's why was it built where it was built? And it was built like in the 1960s, 1950s when they started thinking about this. And the answer to that was, that's how the government thinks, at least the US government, is that if someone, and I'll not say who, drops a nuclear bomb on the White House, this was at the time the radius of the maximal impact. So that's why they built a lot of agencies outside of the Beltway. No one took into account the fact that bombs can, of course, hit elsewhere, and also the radius changes. But in any event, okay, that gives you a feeling of where the University of Maryland College Park is. Since we are, actually, why not? Since we are speaking about group dynamics, I thought that it would be, there's really not much purpose to this movie other than waking you up. But we heard very, very nice talks in the past couple of days. So, for example, we were discussing, there was a question about, do the bacteria, the cells, do they block the oxygen? Can oxygen go? There was, another thing with actually the same talk with traveling waves. And I'll speak about group dynamics of a different kind, but still, whenever I think about group dynamics, I think about this movie this is somewhere in China, and that's a wave pool, and that's not computer animation, that's a real video. And it actually gets better. I think that like a traveling wave. Okay. Yeah. So if you've never watched this, there are a lot of these videos online, and okay. And now we can get to the talk. Okay. So we're done with this part. In fact, let me show you a different movie, and I'll not explain what it is, but I will just say that it was inspired by what we're going to see, the motion of these phototactic bacteria. And all that I would like you to, and ignore for a second the colors. So just think about what you're seeing, and imagine that you see a result of an experiment that looks like that. Okay, this is a simulation. And then you're trying to come up with a mathematical model that describes something that looks like that. Okay, I mean, this is very distracting in the background. Sorry about that. Let me close this thing. Okay, here. Again, ignore the colors for a second. Colors is actually an interesting addition that I played with and never published anything about. That is, let me tell you already what I had in mind with the colors. So think about, I mean, I don't know if you want to think about the disease modeling or opinion modeling or something like that. If you want, these can be members of two parties and they can influence each other under some rules with some probability. Look, I mean, is that what we will call a steady state, a stationary thing? Maybe not. In physics, we would probably call it some like a non-equilibrium statistical mechanics kind of thing to describe that maybe. But it's actually interesting, I think. Oh, no, almost. Let's see. You know, you can watch these things for hours. So, and we actually watched the biological movies for hours. And as I said, that was inspired by that. We'll get in a second to the biology, but just, you know, for you to think about what's going on here. And, ooh, okay, here, it's a blue particle escaped, and no, it infects or something else. Okay, so how would you describe such a thing? And why do we care? Okay, so we'll get back to that in a second, but at least hopefully that intrigues your curiosity enough to sit through the next several minutes of the talk. So I'll tell you a little bit about what phototaxis is. Then I'll tell you about different kinds of approaches to modeling different things that are associated with this phototaxic phenomena. And yeah, there are different kinds of models here, so I'll tell you a little bit about them. And actually what I call the local cell-cell interactions is something that will then result with a movie that I just showed you. So what are we interested in? We are interested in looking at bacteria that use light as their source of energy. They're everywhere. They're important. They're as old as the planet. Most of them live in water, but not only. In fact, they're here on the table and everywhere all the time. And the nice thing about them is, I mean, light is their source of energy. So if they're here now and there is light, well, they'll stay here and they don't care about it. But if I turn off the light in this room and I turn on a light bulb in the corner there or somewhere far away, so there's a little bit of a directionality here, gradually the colony will start migrating in that direction. Okay, that's what you should think about. Okay, so yeah, why do we care about them? I mean, they're super important. They're actually the reason why we have photosynthesis. That's what, so in other words, why we have plants and therefore why we have oxygen to this planet. They're also related to fuel deposits. They are, can be used as fertilizer. In fact, you can actually eat some members of the family. There are others that you should not because they're poisonous. But in any event, what we are wondering about today is the motion of these bacteria and the cyanobacteria, and specifically, I'll be looking at the next slide. These cyanococytes, they are not swimming. They are kind of walking. So how are they walking? What they do is they think about this like I put the picture of Spider-Man there. They send a web and retract. Send a web and retract, and that's what they do. That's how they move, okay? So, you see a lot of models of, like in the mathematical literature, of things that swim. That's not one of these here. They just do something like that. That's the motion. So, why do they move? Because they want to go to the light. Will they make it? Well, I don't know, but that's part of where this group dynamics is. I mean, we don't care really about the individual bacteria, but as a colony, maybe if a colony decides, quote unquote, whatever that means, that it's better to be there, they'll migrate towards light gradually. By the way, they walk quite fast. But before the walking, in terms of the movement, what is clear, and this is light, is that these bacteria, they should identify that there is light. Okay, so there should be photoreceptors on the bacteria. That way they know that there is light. Okay, so we know that there is. The other thing, there should be a way of moving. So from this realization that there is light, there should be a signal that says move. And then what I described about how it moves. I ignored the question of where do you move to? Okay, so which direction to move? Now, I speak with collaborators, biologists side, and they say, well, okay, you know, the bacteria moves in the direction of the gradient of the intensity of the light. They're like, but how would it measure that? Like, I mean, maybe, I don't know. I mean, but what mechanism would that? So is that possible? Is that what's doing? Well, we don't know. Is it moving somehow, jittering and figuring out by that, somehow measuring something? Oh, we don't know. Is it, maybe there are some bacteria that take, for whatever reason, they know how to identify the direction of the light and the others go with them, so different traits. Is that possible? Well, we don't know. But one thing we do know is that this is not instantaneous. I mean, when they start moving, they move fast. So in fact, if you do the experiment and you put them on a plate under a microscope, and so, you know, that distance, within like 24 hours, you would see them dropping off the plate. I mean, they walk quite fast. So you're looking at creatures of, you know, diameter of a micron, okay, and they would walk, you know, 2 centimeters in 24 hours. So do the calculation, but so that's a significant speed that they move in, okay? But it will probably take them 4 hours before they start doing it. Okay, so it's not as if you turn on the light and they instantaneously move in that direction. It takes some time to make a decision, whatever that means, okay? So this is kind of what we want to understand. So specifically, these synechocytes, as I said, the diameter is about a micron. And if you look at the classical experiments that are conducted with these kinds of bacteria, that's pretty much 3 things that normally you would do, at least, you know, maybe 10 years ago. Okay, so one of them, you would take pictures of them under an electronic microscope. So you see here, for example, a division. There's 2 different, you know, technologies for filming. You also see these webs. These are not these pili, okay, that's what they're called, type 4 pili. These are not these things that they use in order to move. But, I mean, you see in the picture something, some medium by which, for example, they can communicate with each other, maybe by sending molecules along these pili, or maybe even mechanical forces. Well, a lot is unknown, but in any event, you see it, okay? Now, one thing you cannot see in that resolution is you can't see them moving because you have to kill them to take a picture with the electronic microscope. So unfortunately, you can't get videos with that kind of resolution, at least using standard techniques. So I don't have such videos to show you, okay? I have different kinds of videos, okay. The other kind of experiment you could do is gene knockout experiments, for instance, and this is a terminology of the biologist. So if the light comes from this direction, the wild type kind is those that when they see the light, they run toward, well, they crawl whatever, towards it. Okay, so they move in that direction. So that's called positive phototaxis. That's the standard thing. Okay, then with this gene knockout, you can create different kinds of mutants. For example, negative phototaxis. So the light turns on and then they move backwards. Okay, or the non-motile one where, you know, there is light and they just go nowhere. So what did you do with this gene knockouts in order to create these mutants? For example, negative phototaxis. Like the non-motile, maybe at this point you somehow disable the photoreceptors so they don't even see the light. That's a possibility, and that will do it. Maybe you ruin the signal transaction. So they see the light but cannot signal to move. Okay, you can do various things, but that's exactly the kind of experiments that I understand are happening on the biological side. While the other thing you can do is you can put them under an optical microscope, and you have to be careful with that. Unfortunately, they do not respond to every wavelength because it's a little bit tricky. In order to put them under an electronic microscope, you need an optical microscope, you need light, but the light kind of is like what you do not want to put there because you want to see how they move. So since they do not respond to every wavelength, you can do that. And let me show you what these kinds of movies look like. So the first movie that I saw when Devaki, my collaborator, approached me, and this was this one. The light comes from here, and you see the individual bacteria, kind of. And then over time, this is just a surface, it's wetted surface. And over time, what you see, and this is ours, you see the emergence of fingering in this direction. So motion starts moving here. Now, we pay attention to these fingers, and that's the direction of the light, but there are other things that are important here. For instance, these guys don't go anywhere. Okay, kind of. I mean, like, why are these moving there and these do not? Well, yeah, but the light is there, right? I mean, this is a play, right? Okay, so it's not like as if they block anything or not, you know. It's just you turn on the light bulb. I mean, that's the experiment, okay? So, and repeat. That's how they were placed. Yeah, they were placed like that. Yeah. So Devaki walks years ago walks into my office and shows me this video and says, "Can you write the mathematical model for that?" So I say, well, I mean, what question are you asking? What do you want me to answer with the model? I mean, I can write models for anything, but what should I try to answer? So the question was, can what we see here be explained as in terms of the behavior of an individual bacteria, or is there some group dynamics involved? I look at this and said, well, that's group. I don't need the mathematical model for that. But I said, why don't you conduct an experiment to answer that? That's easy to conduct. You just put, a few bacteria, you turn on the light bulb. If they don't move, you're done. Right? But that's actually a difficult thing to convince, because, like, I mean, this is kind of a cultural thing, right? So you spend a lot of time and money and do the experiments, and kind of what's the point that you're trying to prove? So, like, if you want to publish a paper, what will it be? I seeded some bacteria on a plate, I turn on the light, and they did not move. Well, for them, that's not a publishable result, right? So, okay, so I said, okay, fine, I'll write the mathematical model, but then I had to buy time, so I said, I can't really see what's happening in the fingers here. The resolution is not good enough. So can you please try to get better movies? Okay, so it took a year. Okay, so then she gave me some time to think. So then there was a whole set of movies. This is not particularly interesting. In this case, the light comes from here. You can see a little bit better the motion, but I mean, like, that's not super interesting. So let me, move to the next movie that is a little bit more interesting. Okay, let me make this one big, because this one I like, because that is important, this particular movie, for a variety of reasons. Again, the light is from the left. Now, several things I want you to pay attention to. One is these guys, and we've seen it yesterday, like, they are not moving. There was not phototaxis that we saw, but these here, they are stationary. In fact, they're stationary until these guys join them. The other thing that's super interesting is what's happening here. If you pay close attention, you'll notice that it's not as if all these particles, they start moving in this direction. Many of them just walk in the opposite direction or do all sorts of weird things. Take a careful look at what's happening here. Oh, more there. I mean, this is not just like, oh, okay, well, yes. It is the same time. Yeah, this is like within the finger. This is actually the finger. This is zoom in on the finger. Yeah, kind of thing. Yes. Yes. The plate. Yes. It's, I don't remember the exact details of how they do it, but it's a wet plate. it is wet. Okay, the surface is lubricated there somehow. I don't think so. No, yeah, it's possible. I actually don't think so. But I mean, again, it's really curious to see what they're doing here. It's interesting. I mean, if you look at what's happening there in the corner, that I'll fast forward that. You'll see here until this point, and then all of a sudden these guys join. Why are they joining now? If you look at some of them even join. It could be that there's some surface tension there, that they feel that something is changing in the surface. There could be all sorts of things. I don't know. Yeah, of course. Light. Yeah, there's nothing special about the light. I mean, these are micron, and you turn a light bulb. There's nothing fancy about that. That's and several hours. Yes. Look at this. Look at so much of this. Yeah. We don't know. I mean, I'll write models and I'll show you how I can simulate things that look similar. But I mean, I don't know. I mean, I'll make some assumptions that I don't even know if they make sense. But this will be observational. So, yeah. Yes. Well, there is. So I'm not. So here you see something where there is a different kind of surface, right? So that's maybe a little bit related to what you're asking. I mean, here's a slow motion version of that. So I mean, basically, ooh, what happened here? Okay, that's a wrong one. Okay, it's kind of a slow motion. Okay, about that moment. I know. I'm just showing you some. They're like. I think that's something with a boundary, and it's not. There's no boundary. It's the surface where they are. So I don't think they produce liquid, but they push it. There is liquid, but there is. It's possible that they create some surface tension around them that changes the thing, and it's possible that the others can also sense it. But I mean, like, why to begin with? There is like something that looks like that. I don't know. Okay. Anyhow, okay. I mean, these are the experiments. So now, I'm being challenged with a question. So I said, well, this is an experiment. Let's try to write something with it. And does it enlighten us in terms of the biology? Well, it creates some questions, sending the experimentalists back to the lab. But in any event, this is the thing. So, I mean, phototaxis, I hear about that. I said, like, well, I don't know. I mean, I'm doing a literature survey. Can't see a single model, or maybe one, of phototaxis. I see a billion models of chemotaxis. I said, well, okay. I mean, there's all that stuff. Why not start with something out there? But I mean, like, so just as a cultural thing, and many in the audience, not everyone, have seen that before. Generally, there are different approaches to writing these things. One of them, of course, is with PDEs. That's a classical way of doing that, in which we, in this case, model two things. One is the food. The other is, in this case, the bacteria. This is a chemotaxis, so they move in the direction of the gradient of the food. So what happens to the food? There is a source of the food, and it's also consumed. That's all in this term. And there's a diffusion term. Okay, so that's the food. So you put food or eat it or diffuse it in the surface. And then what happens to the bacteria, let's say? They can give birth or die. They can diffuse, and they can move. And the speed by which they move is the gradient of the chemical concentration. So this is a starting point, but not very immediately clear how to connect that with what I have been doing, because and there's a lot that has been done in the mathematical literature about that, and I'll not get into any of that. But kind of in my mind, when I'm looking at these things, there are several factors that I want to capture, and I know I could some of them with a chemotaxis model, but at first sight, there are some differences. So one of them is that delayed motion concept, and the other is that, for me, the light source for the bacteria is at infinity. It's not as if I put food here and everyone crawls into it. Here, the movie of chemotaxis, what does chemotaxis look like? No, again, this thing. Sorry. So here is chemotaxis, the classical movie. You put food here and the bacteria crawl in this direction, right? And that actually, for those that don't understand why mathematicians care about it, is the reason is very obvious when you look at this movie, because, I mean, what we do, if we write an agent-based, an individual-based model for these things, no problem. But if we now want to go to a continuum and speak about infinitely many of these, and they all come to this point where the food is, that will create a blow-up, and that is exactly what we do not want to see in the equations. And these are the kind of questions that we ask: do these equations form blow-up? And if yes, what should we do to make them more realistic? Because, of course, the biology does not go to blow up. Okay, does not blow up. So back to here, I'm looking at the 2D thing, no fluid dynamics. I'm seeing different patterns. I want to do something with it. So how do I do it? Well, attempt number one. For attempt number one, I'll make I'll make the, oh, there's any comments I want to make on this slide. Well, we've seen the fingering, we've seen the delayed portion. There is also something that when you stare enough at these movies, you see that the surface is important. So it's important in two ways. One, it looks as if areas that were traveled by these bacteria are easier to travel on, and it can also influence their velocity. And we know different mechanisms by which bacteria do exactly that, like they can consume something and leave something behind and so on and so forth. So you can think about mechanisms of marking the surface, that stuff can diffuse, et cetera, et cetera. The surface is important, okay? Absolutely. It could be. Yeah, absolutely. Yeah. So, and you can sit now and write a lot of different variations of these models. Here are several, okay, and in no particular order. Okay, so one is you can assume that so some assumptions, let's not allow them to give birth or die. I want to describe the surface memory effect, and I am assuming that there is a group dynamics, and I want to encode it. So how do I encode it? The way by which I encode it is I'm saying that, and I also want to encode somehow the delay. I want to say that there is something internal to the bacteria that, think about this as a clock or some gauge or something. I call it excitation. I don't know if it exists biologically, but that helps me explain a delay that has to be built gradually, and only if it crosses a certain critical threshold, then the bacteria will even bother to move. And it's not a question of where to, it's a question of whether it moves or not. And how do I encode this notion of excitation? I say that if the bacteria is here, it has a certain neighborhood that it can sense. Think about some chemotactic signals or mechanical signals or I don't know what, in which neighboring bacteria can provide some information, and by that I can gradually build on this excitation and create and make it move. So the formulation is not super important. What is important are the concepts. There are really three things that I'm describing here. One is the surface. What am I doing for the surface? I'm just marking it. So think about all these. So the bacteria move along these red lines, and I'm marking the red lines on the surface. That's encoded here. The other thing that I'm doing is I'm encoding the excitation of bacteria I call it SI. The excitation, the average excitation in the neighborhood of the bacteria is mu I, and I set it so that the excitation of bacteria I, particle I, points in the direction of the average excitation. So if all its neighbors are really excited, then so it is itself, right? And I am also adding a little bit of a random component to it just to make things a little bit interesting, but also to build the excitation from scratch if I need to. And the other thing is for the position, let me encode that. Well, there's some component. I do not like the Brownian, this is not Brownian motion that we see here. That's not Brownian motion, but I'll put it nevertheless because that's the easiest thing I can put. And then later I'll study what they actually do. And other than that, so that's a noise. Other than that, there is a direction of the light that's encoded here. And there's a velocity. And all this thing says the following. It says, move if the excitation is greater than the critical threshold, and take into account the surface, these red lines, and the gradient of the surface. That's all that's written here. Okay, so I take that, and I know by writing things like that I accounted for the surface, I accounted for the light. I don't know how they know the direction of the light that's encoded here, but there is something that's light dependent. There is a group dynamics, and there is something random in all that stuff. So all that remains at this point is to show you what do simulations of such a model actually look like. There's several technical interesting questions about how you implement it, but let me ignore them first. I'll just say, well, I'll just emphasize one thing. In a model like that, the bacteria are these blue dots. This is not them moving on the lattice. They can go anywhere. Some of these concepts in terms of like how do I mark the surface when they move and all that stuff, I have to think about this, and some of these things are easier to do on an underlying lattice when I implement things. So just a side note. But in any event, they are not moving on the lattice. They'll move later on the lattice, but not yet. So let me show you what such simulations look like. And the first one would be what I would call two dense areas. So now the light is from this direction, and I don't have new particles, and we had this question yesterday. I mean, at a certain point for these things to persist, you need to build more particles, right? They need to, and certainly on the time scale that we're looking at, they do divide. But in any event, but let's look at it again. What happens over time, you see that in it, these I call, what is dense? Dense is really something that it depends on my parameters. Okay, so that's dense. And you see the emergence of these finger-like structures. I can take I'm kind of, what I'm trying to say is, think about this, these red marking as the trajectories. Instead of delta function, imagine that I diffuse them. Okay, so imagine there's something that diffuses. And what I want to say that if you get close to an area, basically, yeah, that they want to go towards it. Yeah, that's, so in some sense, that's not completely different from a chemotactic kind of signal. I mean, but okay, here there's an area where you remember that experiment where said, oh, these guys stay in place and they join only when the others. Well, for me, this is a playing with the parameters to make a distinction between a dense area and a high density area and a low density area. The difference being in terms of the building of the excitations. If these particles are not excited about moving, they'll not move. I mean, all this is a bit of the random jitter there. But as other particles get close to them, they may build their excitation, they move with them, which is exactly what we see in the movie. Again, this is observational. I mean, I'm not saying anything beyond that. Now you can start playing with different scenarios. I mean, you can start with, and unfortunately, I don't have such an experiment, but you can see the emergence of a finger, and if you pay it careful attention, I'll tell you there's even a secondary structure here, okay? So I do not have such an experiment, and you can even ask yourself what happens if you keep changing the direction of the light. okay, and so you can make them walk in different direction and kind of like, you can do all sorts of things like that. So this is all. And by the way, if you do the experiment, you indeed see, like in terms of changing the direction of the light. So if you have light source coming from here and you get this fingering, and now you stop. What is nice about these experiments is much easier than chemotaxis because you can turn on and off the light instantaneously, as opposed to getting rid of the whatever you put there that is still diffusing, right? So you turn off the light and now you turn it on from here and now you get fingering in that direction. Okay, so you can, I don't have movies of that, but this is faster. This is faster. Yeah, okay, right. Okay, now you can do various things. You can write a Foley particle model where you describe the motion and birth and death. You can take the continuum limit. You'll get PDEs. By the way, so these PDEs are basically they are the continuum limit of the initial system that I showed you, the stochastic system, where the structure is, again, quite obvious. I mean, what you get is three equations. One is for the bacteria, the other for the excitation, and the third for the surface. Surface, whatever, bacteria, they move and diffuse. And the excitation diffuses, it moves with the same, the excitation moves together with the bacteria. It's kind of strange that it's separated. You can do it in different ways. You can take it as another variable. In this case, that's not what happens. In other models, it does. And the other thing that does not happen with here, but excitation goes up and down. So in terms of conceptually what's happening there, yeah, you have to allow it to give birth and to die, the excitation out of all things. If you want the bacteria to do that, just add terms here. This is formal derivation. There's actually theory. You can write the theorem and prove it that the solutions in some sense of these two systems are close to each other. Okay. You can also write a kinetic, that's a little bit different. So you can write a kinetic model, and I'm not going to show you simulations of that. Nothing. The velocity is the derivative of the displacement. So now I have to describe two things. One is how the velocity changes, and the other how the excitation is changing. Well, the velocity, there's some cooperative term here, and this is where the trick is. If the excitation increases, it goes over a critical threshold, then the velocity aligns itself with a prescribed velocity. So it's like a Cocker-Smale model with forcing that comes from the external light, and you do something for the excitation. And now you can do all the fluid models and get PDEs. I am not even writing them down. They're different. Okay, so you can do things like that. But in my remaining 5 minutes, I will tell you a little bit about these local cell-cell interactions. That's the first movie, not the swimming pool, but the one after that that I showed you with these blue and red particles. So what's happening here? Well, these are actually, I didn't show you these movies, but they're even with a high resolution. So now you look at the particles in the low density area where I told you, look, the particles are actually moving backward. What are they doing, right? So, well, actually, if you pay attention to them very carefully, you see all sorts of things. For example, there are particles that just don't move. Then there are particles that move as pairs. Then there are particles, and that is the interesting one, that move towards neighbors. So, and on top of it, of course, there's forcing in the direction of the light. So we sit down and say, how do we get a model that does exactly, that describes what these particles are doing? So this is it, okay? Here's what's happening here. I call it the people in a party kind of model, okay? So think about this like that. Let's say we are all in a party, but we don't sit on tables. We stand in different parts of the room. And then I'm standing here, and I'm just standing, okay? And now I decide that, yeah, I'm kind of, lonely, or whatever. I want to socialize, or I don't want just to stand like that and look weird. So I see Christian. So I said, well, okay. So I start walking in the direction of Christian. Now, of course, Christian sees me. He can do one of the following two things. He can, so Doron is walking towards me. I don't want to speak with him. So okay, I'll go there. Okay, now I'm going to play it cool because, like, what are my options? I can start stalking Christian, but that will not be nice, right? So I said, well, okay, I just pretend as if I'm walking to somewhere else that's in that area. So I keep walking, or maybe I stop, or maybe I change the direction. Yeah, okay, I see Marie there, so I say, well, I'll go towards Marie, okay, and so on and so forth. So that what's happening here really is an interesting motion, and that's actually the motion that you saw in the first movie. Second movie. There are two independent decisions here. This is a model with memory. So particles either move or not. So I am standing or not. With some probability I move, with some other probability, or not. Okay. Then if I move, I have two options. One is I move in the previous direction that I can. So if I'm here, so I probably walk like that to come here. So now I stopped, and now I'm restarting. One option is to continue moving in the same direction. The other option is to look around and see if there's any neighbors and move in the direction of one of them. Not mean field, not average, not any, one of them. That's what's written here. That's it. Okay. So what does that look like? Well, let me show you the movie. So this is what a movie like that looks like. Well, what you see here is the distance, the diameter of a neighborhood, and they're confined to a circle in the simulation. Okay? So that's one option. Just leave it to that size. Here is another one where the diameter is bigger. And what you see is that you gradually create some colonies here. These groupings gradually over time. In fact, if I go back here, what you see here, you have, again, just a longer time, 250 particles. The diameter is exactly what's represented here. Different realization. It's a random process, but there's something similar about these images. Okay. But these images, that's a different model. These images, and now you can do it in R2 or with periodic boundary conditions, so on and so forth. In fact, what you can do is then ask yourself what kind of trajectories these particles do as they move. Okay, and you can study that and do whatever you want with that. And let's see. So, and now you can ask what happens if there's some preferred direction, okay? And like a light coming from here on top of it. So will I get a migration towards the light? You can impose these things. Then you can ask questions. What happens if some of these particles can sense the light and others do not? Will such a motion move everyone or just a group of those that sense the light? And so on and so forth. In fact, this motion is so interesting that after writing this 2D version, what we did was we went and, why did it leave the thing? Full screen. Okay. We said, well, actually, it's interesting. Why do we have the particles move in the space? Now let's do some math with it. So let's allow them to move on a line. And what happens then? You can move to the right or to the left. You can stop or not. The decision whether to move to the right or to the left will be a function of your neighbors. Okay, the number of the neighbors. You can weight the probabilities in such a way. You can actually write ODEs to do that. And you get examples of exactly that thing of you have groups, but the beautiful thing here is you have a lot of models of aggregations in math, but there are not that many models where aggregates break down, or there's mechanisms to allow them to break down. So I have here a group of particles, they move like that. I have here another group of particles, but if a particle here all of a sudden decides to escape, we'll join this group, and you can do, so there's nothing that keeps them really, other than with some probability they should stay there. You can then go and write these models on a 2D lattice and allow them to move as on a 2D lattice, and all sorts of interesting things can happen then. For example, you start with particles. They are on 2D lattice. Some particles move up and down, left and right, and whatever. Over time, they join into these groups. That's at a later time. In a later time, there's some particles that stayed here. If you wait long enough, they'll be here. That's not necessarily what happens. You can wait the same time and you get a completely different picture. I mean, yeah, but in any event, these are a little bit difficult to see, but here you see a little bit better, like what you get some strange, like, distributions of particles, and they move in these different directions and so on and so forth, which brings me to the very last thing, which is the very first thing I showed you, and that is that movie, not this one. Yeah, that movie here. Go away, this thing. Okay, sorry. That movie here of, this is actually what this motion looks like. I put here a large neighborhood, and blue and red, I mean, that's an addition on top of that. Now you can play with it in many different ways. You can say, well, this is maybe a, you know, I mean, I didn't write, I try to avoid working on COVID or anything, but if you want to have like something with space and disease propagation, things like that, then based on certain rules and what have you, I mean, like, this will not describe the life in the city, but, you know, in a small group of people, it can maybe a relevant model. And I don't know a lot of them that actually captures things that are like that. There are some statistical things, there are some other things. In any event, with this, I am going to thank you for your attention and end here. Thank you so much.